Hello everyone. Today I will talk about principal component analysis, also known as PCA. To start, we'll wonder why and when do we perform PCA and what problem does PCA solve? PCA is particularly useful when we have to deal with big dimensional spaces with a high number of variables. If we have n individuals described by 2 or 3 features, we can directly visualize the data and interpret it. But what happens when we have a much larger number of variables? In this case, PCA can help us reducing the number of dimensions. This could help us to perform exploratory analysis, visualize the data and its structure. It can also help us to perform feature selection tasks, identify the features varying the most across the samples, and select a reduced number of independent features, for example to perform other techniques that cannot handle too many features. What is the principle of PCA? How does PCA actually reduce dimension? PCA is an unsupervised technique which looks for a series of principal components, which are linear combinations of variables, such that they maximize the variance and such that they are orthogonal, so independent from each other. We will illustrate an example on a 2D space for comprehension's sake, but note that applying PCA in 2D spaces isn't usually very useful. There we have a cloud of points representing individuals described by 2 variables, gene1 and gene2. We look for the linear component maximizing the variance of this cloud and get PC1, which can be written as a linear combination of gene1 and gene2. Next, we can repeat the same reasoning for principal component 2, but knowing that it must be orthogonal to principal component 1, and this is what we get. Before applying PCA to our data, it is important to center and to scale it, especially when variables have different units. Then, we can compute the correlation matrix and apply basic algebraic operations to finally get in return a list of principal components and their associated percentage of explained variance. Principal components are always ranked according to this percentage. The first principal component has the highest percentage of explained variance, and so on. Now we will have a look at the parameters and the results interpretation. So there's good news. There are no parameters to choose, which make PCA highly reproducible. However, there's still some choices to make, and the main one could be how many principal components do we want to keep? There is no right answer, but here are a couple of possibilities. We can wonder how many principal components do we need? Or for instance, how much variance do we want to keep? And finally, how many principal components have a big explained variance? For instance, we can see on the right that the first 3 principal components have a high percentage of explained variance compared to the others. This could help us set the threshold. Another aspect of PCA is that it allows to see the correlation between the variables and the principal components. We can therefore estimate how well a variable is represented in the reduced dimensional space of principal components and estimate what kind of information a principal component carries in terms of variables. On the right, you can see a graph representing each variable as a vector whose coordinates in the reduced dimensional space are its correlation coefficients with the considered principal components. Variables with long vectors, so close to the circumference of the circle, are well represented in this space, while variables close to the center, like Fe, are not. In this example, we can see that the variable BOD is strongly positively correlated with principal component 1, while the variable temperature is correlated with principal component 2. Now we can look at the individual's projections on the principal component space. As previously announced, this helps us visualize the data, looking at its structure, identifying subgroups, possible outliers or tendencies. Here on the left, we can see that the data clearly splits into two subgroups. On the right example, the data has been labeled, and PCA shows that there is a clear separation between desserts on the one side and starters with main dishes on the other side. To conclude, I will talk about the main limitations of this approach. First, it assumes and detects linear relationships in the data. Secondly, it assumes Gaussian distributions of the data. Next, it is quite sensitive to outliers that can lead to disruptive principal components. Therefore, if some outliers are found, it could be useful to exclude them and repeat the analysis. Finally, PCA does not conserve small pairwise distances when going from the original space to the principal component space. Thank you all for your attention.